The difference between plant and bacterial photosynthesis. In bacteria, there is no chloroplast present, and chlorophyll is dispersed in the cytoplasm. In plant, the chlorophyll is found in chloroplast. Now you can see second comparison, that is produces O2. Plant does produce O2 because they utilize H2O. The oxygen is actually arises from the water molecule. In bacterial photosynthesis, oxygen is not produced as they doesn't utilize H2O, but utilize H2S, hydrogen sulfide. And the type of chlorophyll is different too. You can see the chlorophyll used by wo- plant absorbs 650 to 750 wavelength light, nanometer wavelength light, but bacterial chlorophylls absorb 800 to 1000 nanometer of light. In bacteria only one photosystem is present. Uh, in plant there is two. You can see the photosynthetic electron donor is H2O, and that is H2S in bacteria. Now this is a plant PS1 and PS2, that is whole cyclic and non-cyclic. This is shown in cyclic phosphorylation. In non-cyclic phosphorylation, where this all summarize the light independent reactions. First, when the light strikes the photosystem, it excites the electrons. And the oxygen is broken up from what H2O, and two hydrogen are released. Two hydrogen proton are released. Now, the primary acceptor is ferredoxin. The ferredoxin transport this electron through electron transporter. This also initiates chemosynthesis of ATP, provides energy for the use of ATP synthesis. Now, this electron terminal continues to photosystem P700, and another light dependent reaction excite UV ray excite photosystem 1, causes again release of two electrons. Because again at P700 the two electrons are prevailed from here and continue to move here, but the energy here was utilized to form ATP and was lost. At P700 there is now another excitation is gained by light, and the two electrons move here. There is a plastoquinone. Uh, it is plastoquinone cyanin is present there. Now this electron moves to NADPH and attaches to NADP and attaches to hydrogen atom because of the energy and form NADPH. This NADPH and ATP moves to Calvin cycle. In cyclic formation of bacteria, sulfur bacteria, only one photosystem unit, that is P840. In this photosystem, a similar pattern is then the proton is striked onto the photosystem and the photosystem transfer into reaction center. Now the reaction center, from the reaction center is on to electron acceptor, and from this complex ATP is produced and chemosynthesis of ATP is done. Another electron acceptor accepts this and transfer it to the photosystem 1, that is reaction center P840. That is an now it generates ATP via electron transport that was similar to that of plant, and there is an oxygenic photosynthesis. That means this doesn't contain oxygen. And that's already explained that electron positive electron is there, and that generates a proton needed for 80%